Universidade de São Paulo in Brazil, but the work I'm going to present today was done at the University of Cambridge when I was working with French chickens, and this work was also done in Chuan cow. This is the first of our coevolution section today, and I hope I can convince you that Drosophila is a great system to understand coevolution, and especially to understand the mechanisms that are important for coevolutionary dynamics. So, in almost all population of organisms are attacked for a variety of parasites and pathogens, and a striking fact about that is that we have a lot of variation on how the individuals resist to an infection. So there's a lot of variation in populations for resistance to infection. And we don't know much about what are the mechanisms that keep this variation in population, and understanding this is important to understand coevolutionary dynamics. And the first step to understand this, why this variation exists in populations, is to understand the genetic architecture of the prey. That means we need to understand the number of genes involved, their effect size, and their interactions. And a great system to answer this question is the interaction between Drosophila melanogaster and its viruses. So in Drosophila, we can do control and highly replicate experiments. And a second advantage is that we have this great resource for genetic mapping. So today I'm going to talk about two viruses that attack Drosophila in the field. So these are two of the main viruses of Drosophila. So we have DCV, Drosophila C virus, and the Sigma virus. So DCV is a highly virulent virus. It is horizontally transmitted, and it kills a fly in just a few days. The Sigma virus is a little bit less virulent. It's vertically transmitted, but it causes about 20% reduction in the fitness of the fly. In a previous study done by Mike McGuire and some co-workers at Frank Chicken's lab, they did a genome-wide association study to map genes associated with resistance to these two viruses. And for the Sigma virus, they found two major effect genes that explain 37% of the genetic variation in the population they studied. For DCV, they found a single major effect gene that called pastoral, and this single gene explained 47% of the genetic variation in resistance to DCV. But this study was really successful in mapping this gene, explaining a large amount of genetic variation, but there is still a large amount of genetic variation that was not explained, right? And this is the common problem of the missing heritability, or our inability to identify the genes responsible for some of the genetic variation present in any trait. So the study I'm presenting today, we use a different mapping panel. This is the DSPR, the Drosophila Synthetic Population Resource, and this is how this panel was developed. So they have two panels, panel A and panel B, and each panel we started with 8 founder lines. So these were inbred lines. They have their complete genome sequence, and then what they did is for each panel they outcrossed these 8 inbred lines in a population for 50 generations. So during these 50 generations, you shuffle the genome of the different founders, right? And then they developed more than 800 inbred lines for each panel. And for each recombinant inbred lines, they did red sequencing, so they were able to identify for each segment of the genome of these inbred lines which founder they inherit that segment from, right? And one of the advantages of using this approach, this panel of flies for mapping, is that you have much higher statistical power. So we are not limited as in the previous study when we were limited to just finding major common effect loci. And with this additional power, we can also look for epistasis and how some of these loci may interact with each other. So here's what we did. We did 2 separate experiments. One experiment for the Sigma virus, another experiment for the Drosophila C virus. And for each experiment, we infected more than 800 lines. For each line, we had 20 flies that were infected with the virus. So we infected loads of flies, more than 26,000 flies. And here's what we found. So we found a lot of genetic variation in resistance to these 2 viruses. So here I'm showing the results for about 100 recombinant inbred lines that have more than 2 biological replicates. So this is the results for DCV. In each bar here is a different line. And as you can see, there's a lot of variation among the lines. So for some lines, the flies die about 2 days or 3 days after being infected with the virus, while some mice are really resistant, they can survive for almost 15 days after being infected, right? And we also found out, and for DCV, we found that about 6-7% of the variation that you observe in our phenotype is explained by the recombinant bred line. So this is the genetic variation in the population. For the sigma virus, we also found a lot of genetic variation. So here what I'm showing is the proportion of flies that become infected with the sigma virus 13 days after being injected with the virus, right? And again, you can see that some mice are really resistant, about none of the flies become infected, while some mice are super susceptible. Almost 100% of the flies becoming infected, right? And we estimate that about 70% of the variation is genetic. It's explained by the recombinant bred line. Then we did our QTL analysis to map the loss size associated with resistance. And when we did that for DCV, first we found this major peak on the left arm of the second chromosome. That peak defined a 4 kb region with 9 genes. One of the genes was pastoral, right? So this is a gene that was previously associated with resistance to DCV. So we are doing something right here, but we're not getting anything new. It's the same old gene. So what we did after that is to repeat the analysis using the genotype of the gene pastoral as a covariate. So we are looking for associations with DCV resistance, controlling for the effect of pastoral. And when we did that, we find 2 more peaks. One, highly significant peak on the second chromosome, right arm of the second chromosome, that defined a 30 kb region with only 2 genes. And we also found a minor peak here that defined a 36 kb region with 65 genes. The next thing we did was to look at the alleles of these different PTLs. So for each of the founder alleles, we assigned them to the 2 categories. One is the susceptible category, and the other one is the resistant ones. And for the first QTL, the pastoral QTL, we find a clear division with 5 of the founders carrying the susceptible allele, and these flies here, usually they die 5 days before the flies that have carried the resistant alleles. So the effect size of this QTL is about 5 days. And this single QTL explains almost 80% of the variation in the population. So it explains a lot of the variation. We did the same for the other 2 QTLs. For this second QTL, we have just one allele that is the resistant one. All the other founders have the susceptible allele. Only one that's susceptible. All the others have the resistant alleles. And the effect size of this was 2.5 days, and it explains 11% of the variation. So together, these 3 QTLs explain more than 90% of the genetic variation in the population, and we can conclude that resistance to DCV has a very simple genetic architecture. So it works in a near Mendelian way. So 2 major effects alleles that explains a lot of the genetic variation, right? And then we did the same for the sigma virus. And here the story is a little bit more complicated. So for the sigma virus again, we did the QTL analysis and we found a major peak that defined a region of 38 kb with 34 genes, and that region includes REF2P. Again, this is a gene that was previously known to be associated with resistance to the sigma virus. The same old gene again. But when we repeat the analysis using REF2P as a covariate, we found 4 additional peaks. And these peaks range in size from 140 to 890 kb and contain from 23 to 103 genes. Again, for each of the QTLs, we divide them in the founder alleles in resistant and susceptible allelic class. We calculate the effect size and the variance explained by each of them. So the most important QTL here was the REF2P QTL. This one explains 24% of the genetic variation in the population. And the founders could be divided in 2 resistant alleles, and all the others have susceptible alleles, right? The effect size for REF2P is 0.31. That means that the difference in the proportion of flies that become infected after being injected with this virus has a difference between 0.31 between the resistant group and the susceptible group. So it's a huge effect size, right? For the other QTL QTLs, you also have a large effect size. It varies from 0.11 to 0.38. And each of these QTL explains an additional amount of the genetic variation. It varies from 3.5 to up to 6%. So for sigma virus, the genetic architecture was a little bit more complicated than for the first virus. So we have 5 different alleles that explain with varying effects. And together, these 5 QTLs explain 42% of the genetic variation. So there is still some missing heritability, right? There is still some part of the genetic variation that's not explained by the major QTLs we were able to identify. And we have some evidence that this may be caused by a loci of small effects. We know that in our population, major effect loci that are rare would be pushed to intermediate frequency because we just started denying founders, right? And we have some evidence as well that epistasis is not important, and I'm going to show some of the results for the epistasis right now. So we took 2 approaches to look for epistasis among the different loci we identified. So the first one was to look for pairwise epistasis between all the loci that we identified in the first part. So the 3 QTLs associated with DCV resistance and the 5 QTLs associated with sigma virus resistance. So what I'm showing here is for each pair of these QTLs, I'm showing the effect size, major effect size of the first QTL, second QTL, and the interaction between the 2. And what you can see here is that the interaction term is not significant for any of the pairwise comparisons. So we can conclude that there is no major epistasis between these identified loci. But we also took a second approach because identifying QTLs that have epistatic effects, just looking for the main effect may be really hard because the effect on the phenotype may vary depending on the genetic background, right? So we have a really hard time to map them. So what we did was to look for modifiers of the QTLs that we identified in the first part. So we rerun the genomic scans using the QTL we identified in the first part as a covariate, and we are looking for interactions between that genotype and the genotype of the recombinant bread line. We did it for all our 8 QTLs. For 7 of them, we didn't find anything interesting. But for one of them, we find a peak that's a modifier. So for one of the sigma QTL that is related to resistance to the sigma virus, we find one peak on the right arm of the 30 chromosome that modifies the effect of this QTL that we have identified before. So here I'm showing the resistance susceptible allele of this QTL we have identified, and here alleles A and B of this modifier. As you can see, this locus modifies the effect of the previous identified QTL. So just to summarize what we found extensive genetic variation resistance to both virus. For DCV, for Drosophila virus, we found a very simple genetic architecture with 2 major loci that explain 90% of the genetic variation. For sigma virus, we found 5 loci of varying effect. It explains 42% of the variation. And even though we were able to map one modifier locus, in general, its status was not very important. So I want to thank these people and these institutions for giving us money. And if you're interested in doing evolutionary biology in Brazil, just look at my lab link and we can talk about this more. And we may have time for one short question.